Greetings and hello. My name is NICOL and Harry, metric number BM241100068, and I'll be presenting my SSM topic, which is about pediatric geneticist. My presentation has four contents, and one of them include the anatomical differences between an adult and a child, the components and functions of a gene, the effects due to gene mutation or defect and treatment, and a specialist from field of interest. My first content, the anatomical differences between an adult and a child. Let's first start with the head. Now we can see two pictures here. One is of a child's skull and the second one is of an adult's skull. As you can see, an adult's skull does not contain fontanelles. Meanwhile, a child's skull does, such as the sphenoidal quad fontanelles, mastoid fontanelles, posterior and anterior fontanelles. The adult skull doesn't contain fontanelles because they have already fused, leaving behind sutures, which are immovable joints called fibrous joints that protects the brain from injuries. This is why a child's head is much more fragile compared to an adult's, because their sutures hasn't fused yet, making their skull vulnerable towards external forces and possibly greatly injuring the brain. Next, we have the skin. Now, we can see that an adult skin has slower rate of water loss, meanwhile a baby's skin has faster rate of water loss. This makes the baby skin more prone to dehydration and evaporation of water. Furthermore, the adult skin has thicker stratum corneum and thicker epidermis compared to the baby skin. This makes the baby skin more prone to infections. Next, we have the GI system. In a child, the tongue is large and the nasal and oral passages are small. In an adult, the tongue is small and it has large nasal and oral passages. Inside a child, the stomach, specifically the cardiac sphincter, is relaxed. Meanwhile, in an adult, the cardiac sphincter is tight. In a child, the stomach empties every 2 to 3 hours because food passes very rapidly, producing loose stool. While in an adult, the stomach empties very slow because passes slowly due to absorption and it produces hard stool. Inside a child, the liver is 4% of total body weight and occupies more space in abdominal cavity, while in an adult, it only occupies a little bit of space by the liver. Moving on to the respiratory system, in a child, the respiratory tract is very delicate and does not produce mucus. In an adult, it's well developed. In a child, the eustachian tube is short, straight, and in short distance between the trachea and the bronchi, whereas in an adult, the eustachian tube is long. In a child, the tonsils and adenoids are large, involved in production of antibodies, where in an adult, it has smaller tonsils. In a child, the respiratory tract obstruction occurs more commonly due to short airway, and in an adult, the less chances of obstruction occur due to longer airways. Lastly, in a child, the respiratory rate is more, while in adult it's less. For the cardiovascular system, in a child the heart sounds are high pitched, in an adult the heart sounds are low pitched. The pulse rate for a child is about 140 beats per minute, while in an adult the pulse rate is about 80 to 90 beats per minute, and the blood pressure is lesser in a child than an adult, which are 100 over 60 millimeter mercury, whereas in an adult, it's 120 over 80 millimeter mercury. In the hematologic system, a child's blood volume is equals to 85 milliliter per kilogram of body weight, whereas in an adult, the blood volume is equals to about 60 to 70 milliliter per kilogram of body weight. And in a child, the lifespan of the red blood cells is around 60 to 90 days, whereas in an adult, it has around 100 to 120 days of lifespan. And the newborn RBCs are microcytic, while in an adult, the newborn RBCs are macrocytic. For the urinary system, infants and toddlers don't have control over their urination, while in adult they have. The bladder capacity in a child is 2 less, while in adult they have about 500 milliliter. And the urinary system in a child is influenced by stress, whereas in adult they are not influenced by stress. Moving on to the endocrine system, in a child the production of growth hormone is high, especially in pubescent girls, whereas in adult it's slow or restricted. The adrenal glands are small and have limited functions, whereas in adult the adrenal glands are normal. And in a child they lack homeostasis control, while in adult their homeostasis are normal. In the reproductive system, I'm only going to talk about the female and male child, because we all know how the female and male adults' reproductive system work. So in a female child, the ovaries are approximately 100 millimeter in length, and 2.4 millimeter in width, while in a male, the testes in a male child are 1.5 to 2 centimeters long and 0.7 to 1 centimeters wide. In a female, the functioning of ovary starts at age 12 to 13 years, whereas in a male, the size of testes increases gradually and reaches maturity between 13 to 17 years. And lastly, in a female child, the faulty development of ovaries lead to precocious puberty or delayed puberty. Moving on to the musculoskeletal system. In a child, the bones of the face and cranium are ossified, but other bones develops up to adolescent period, whereas in an adult, all of the bones are ossified. In a child, the bones are still growing. In an adult, the bones have stopped growing. In a child, the muscles are 25% of weight, with muscles and ligaments being very weak, whereas in an adult, the muscles consist of 40% of the body weight. Lastly, we have the nervous system. In a child, the brain is around 300 to 500 grams, while in an adult, the brain weighs around 1,300 to 1,400 grams. In a child, 90% of the brain grows until 2 years of age, whereas in an adult, the brain is fully developed. In a child, the reflexes activities are present during infancy, while in adult, the reflexes activities are present during adulthood. My next content is the components of a gene. This is the general structure of a gene. It has a lot of components, but each components are important for the function of a gene. For the first one, we have the promoter, which is a place where cellular machinery gather to start building an RNA message. Protein binds specific DNA sequences on the promoter and recruit RNA polymerase. Once RNA polymerase attaches, transcription begins. Next component is the stop codon, which is made up of the bases ATG or AUG on the RNA molecule, which codes for the amino acid methionine. It tells the ribosome, the cell's protein building machine, where to start reading to build protein. Next is the switch, aka enhances and represses, or regulatory DNA. This switch controls when, where, and how much RNA is made. Switch proteins bind to specific nucleotide sequences in switches. The DNA molecule folds, bringing switch proteins into contact with the promoter. Because the DNA folds, switches can be far away from the promoter state regulate. Enhances enable or upregulate transcription. Represses disable or downregulate transcription. Next one, we have the exons, which are connected or spliced together to form the mature mRNA. Together, the exons make up the gene's protein coding region. By putting different combinations of exons together, alternative splicing allows slightly different proteins called isoforms to be made from the same gene. Next one, the intron. Introns are cut out when the exons are pasted together. But introns aren't just junk. Embedded within introns can be switches, separate protein coding regions, or non-coding RNA molecules that regulate gene expression. Humans have an average of about 8 introns per gene, but not all genes have introns. Next, we have the protein coding region, which is nucleotides specify the order of amino acids that make up a particular protein. A single-stranded copy of the protein coding region is sent to the ribosome. The ribosome uses the information to build a protein, a process called translation. And lastly, we have the stop codon, which are bases UAG, UAA, and UGA. They can all be stop codons. They tell the ribosome, the cell's protein building machine, where the protein coding region ends. A gene is very important towards the human body, and it has a lot of functions. Some of them include building structures, where genes encode proteins that build muscles, bones, cartilage, and other internal structures. It can also direct bodily functions, where genes encode proteins that direct the body to do things like grow hair or flex muscles. It also supports bodily functions because genes encode proteins that indirectly support bodily functions, such as helping the immune system or blood flow. And lastly, it influences the physical characteristics. For example, genes directly influence physical characteristics like hair color, eye color, and physical quirks. We have already learned the components of a gene and the functions of a gene. So, what happens when a gene defect or gene mutation occurs? When a gene mutation or a gene defect occurs, it can affect the human's whole body. But I'm just going to focus on one part of the human body, which is the cardiovascular system. So, what is the effect of gene mutation or defect towards a child's cardiovascular system? Here, I have six, which are vascular abnormalities, metabolic and genetic disorders, syndromic conditions, congenital heart defects, cardiomyopathies, and arrhythmias. Among all the effects, I'm also going to focus on one effect, which is a syndromic condition. Now, as you can see in the picture here, it is a baby that has Down syndrome. Down syndrome is a genetic condition where a person is born with an extra copy of chromosome 21. This means that they have a total of 47 chromosomes instead of 46. Here are more pictures that showcase how a baby or how a child looks like with Down syndrome. And if you notice, their features are very similar and they look almost identical to each other even though they're from different parents. So how do how does Down syndrome occur? What causes it? To answer that question, Down syndrome is caused by an error in cell division during the formation of an egg or sperm cell, also known as non-disjunction. This results in a sperm or egg cell with two copies of chromosome 21 instead of one. When this cell fertilizes another cell, the resulting baby will have three copies of chromosome 21 or trisomy 21, which is shown here in the diagram. The extra genetic material from Down syndrome disrupts normal development and is associated with physical, cognitive, and medical challenges, including congenital heart defects. Up to 50% of individuals with Down syndrome are born with congenital heart defects, the most common being atrioventricular septal defects (AVSD). So, what are AVSD? AVSD are structural defects in the heart where the walls separating the chambers, atria, and ventricles, and the valves controlling blood flow, which are the tricuspid and mitral valves, are malformed. These defects result in a single large hole in the center of the heart and improperly formed valves. As you can see here in the picture, there is a huge hole in the center of the heart. So, how does this affect the heart? Blood flows abnormally between the left and the right sides of the heart, causing increased pulmonary blood flow, which causes overloading of the lungs with blood. Then, the volume overload in the heart, leading to an enlarged heart and eventual heart failure if untreated. And in severe cases, oxygen poor blood mixes with oxygen rich blood, causing bluish discoloration of the skin known as cyanosis. So, what are the treatments that can be done to treat AVSD? Number one, if the cases are mild, medical management can be done. Medications such as diuretics or ACE inhibitors are taken to help manage symptoms of heart failure temporarily. Or, in much more severe cases, a surgical repair is needed. Surgery is typically performed within the first 6 months of life for complete AVSDs or when symptoms worsen for partial AVSDs. Involves closing the septal defect with patches and reconstructing or repairing the AV valves. With timely surgery, most children have good outcomes, but lifelong follow-up is necessary to monitor for complications like valve dysfunction or arrhythmias. The last content of my presentation is the specialist in my field of interest. First, we have Dr. Lok Hok Nul from Kuala Lumpur Hospital, Malaysia. He took MBBS in Malaysia, MRCP in UK, and fellowship in clinical genetics in Malaysia. He is a consultant clinical geneticist and pediatrician and a clinical lead in inherited metabolic diseases at the Genetics Department of Hospital Kuala Lumpur. We also have Prof. Dr. Tong Miao Kiong. He studied medicine and trained in pediatrics at the University of Malaya. He is a fellow in clinical genetics at the Women's and Children's Hospital Adelaide. He is also a senior fellow at the renowned Murdoch Children's Research Institute in Melbourne, Australia, and he is recognized as the first board certified clinical geneticist in Malaysia and helped establish clinical genetics as a subspecialty in the National Specialist Register, Malaysia. Thank you very much. That is all from me. I apologize sincerely for any inconveniences and also any inaccurate information provided, if any. But overall, I hope the presentation was up to the standards, and thank you very much.